Hi everyone. I will be talking about the pre-metastatic immune niche in the tumor surrounding lymph nodes of non-small cell lung cancer patients using multimodal and spatial single-cell profiling. So the immune microenvironments in non-small cell lung cancer and the surrounding lymph nodes are associated with the clinical stage and outcomes. However, the immune microenvironment in the stage 1 non-small cell lung cancers that particularly are associated with the metastasis negative lymph nodes have not been fully described. So we use single cell profiling and spatial profiling to identify the immune subtypes and their spatial interactions in the lymph nodes that surrounds the tumor. And our aim is to propose how various immune niches may relate to the non-small cell lung cancer's progression and severity. So the methods we use, as I mentioned before, is to take fresh samples from the patient's lymph nodes using fine needle aspiration, as well as obtaining these tumor samples from the lungs, and then do single cell sequencing with CTSI, which are antibodies, a cocktail of antibodies that identify the cell surface proteins. We put in through sequencing and we analyze the results. Simultaneously, we also obtain FFPE sections of these lymph nodes and tumor samples, and we do imaging mass cytometry, which we can use up to 42 different antibodies to create a panel to spatially resolve the immune microenvironment within the tumor and the lymph nodes. Our samples are very well obtained because we get both from the fine needle aspirations as well as FFPE tissues that are directly from the patients. So we guarantee that the tissues are fresh. So in total, we have 10 needle biopsies of these FFPE tissues collected from 10 different treatment-naive patients suspected of early-stage lung cancer that are undergoing surgery at BMC. In total, we obtained 76,000 cells, and utilizing the resected tissue from the same case, we did H&E staining and multiplex immunofluorescence first at these regions so that we can select the ROIs for imaging mass cytometry using our 42 antibody panel that we've developed in lab. So the results from the CytSeq portion is that first we get the broad immune cell types, which we can identify the B cells, myeloid cells, TNK cells, but also, just for example here, within the T cell population, we can identify many more of the finer T cell populations using the RNA and also the cell surface protein expressions. From the FFPE tissue, from the imaging mass cytometry, we can do something that's similar to get the broad cell type populations, and what's not shown here is the finer cell subtypes. So here showing in the CytSeq are the different CD8 T cells, CD4 T cells, and dendritic cells that are shown. And here in the first top half, you can see the proportions of the immune cells that are enriched in the lymph node or in the tumor. We can also see individually which cells came from which patients, if they came from which patient's lymph node or tumor, as well as we know the tumor stages of the patients. Here we have the RNA expression and also the protein expression of each of these finer cell clusters. One interesting thing is that within the dysfunctional CD8 T cells, we can see the majority of them are in the tumor, but we can also see that some of them are also in the lymph node, which is interesting. So we wonder if these originate from the lymph node or is they exited the tumors and traveled to the lymph node. Another interesting cell populations that I want to show here is the mreg DCs and the Tregs. So in total, the M1 lymph nodes, which are the lymph nodes that are closer to the tumor, have more of these immune cell types compared to the low stage patients of M1 and N2 patients. So the M1 nodal stations of these increased staged patients have more of these dysfunctional CD8 T cells, Tregs, and mreg DCs. We suspect that from the CTSI data that these cell types may play a role in the high stage of the patients, the progressiveness, and the severity of the cancer. So using imaging mass cytometry in the IMC data, we want to see how these cells interact each other, whether these dysfunctional CD8 T cells are interacting with any of these cells, and how they might have appeared in the lymph nodes. So in the IMC, what we did after we've identified the finer cell populations, we use a neighborhood analysis where we expanded a certain distance, so in this case 20 micrometer, from the cell's center to identify what cells are in the vicinity of the cells. And using this approach, we've developed these immune niches, immune spatial niches. And then here in the figure 3b here, we're showing that these niches here, what they've identified are the B cell follicles. So the each of the individual niches are composed of different kinds of immune cells. Due to their spatial proximity, we make them into these spatial niches. And this B cell follicle here shows that what these immune subtypes are. And then after we've successfully identified these immune niches, we want to see which immune niches are enriched in the high stage or increased stage M1 lymph nodes. And we've identified three immune niches that are more enriched in the M1 lymph nodes compared to the other lymph nodes. And these in the yellow here, which we're not going to mention too much, is that they're going to be less enriched in the increased stages. So from the H&E, we did the IMC, and here we've identified the different cell populations. And in the niche 39 here, we see the appearance of these mreg DCs, CD4s, which are the Tregs here, and then the CD8s. From the marker expression, it matches exactly what we see in our CTSI data, where the mreg DCs are these TIM3 positive mreg DCs, as well as CD4-4, which are the Tregs, and the CD8-5, which are these dysfunctional CD8 T cells, based on the protein expression. So here we see that the niche 39 are composed of these different immune cells interacting with each other and are enriched in the higher stage M1 lymph nodes. So what we propose, the hypothesis, is that with these mreg DC5s present, they form this spatial niche that are indicative of these more severe non-small cell lung cancers within the lymph nodes of those patients. And perhaps this could be a spatial biomarker that is suggestive of the patient's progressiveness and severity. So for the future directions, we want to expand the number of patients that we recruit so that we can obtain even more statistical significance for our spatial biomarker that identifies these lymph nodes, spatial immune microenvironment that may be indicative of higher stage non-small cell lung cancer progression. Thank you.